The first phase of a cell's life is known as the G1 phase. During the G1 phase, the cell grows larger and is metabolically active. It is a period of intense growth and activity. In this phase, the cell prepares itself for DNA replication, which is also known as the synthesis phase. In the G1 phase, the cell goes through the following check. Is the DNA okay? Are there enough resources for the DNA to replicate and build extra proteins? Is the environment okay for the cells to divide? Now moving on to the S phase, which is the synthesis phase. The nuclear DNA replicates. Let's zoom into the cell. The cell contains 23 pairs of chromosomes. Each chromosome has only one chromatid. Hence, the cell has 46 chromosomes and 46 chromatids. The DNA undergoes replication, which leads to the doubling of the DNA per cell. At the end of the S phase, the cell still contains 23 pairs of chromosomes, but it has 92 pairs of chromatids. Hence, there is no increase in the chromosome number, but an increase in the chromatin number. The S phase is followed by the G2 phase, which involves the synthesis of proteins required for mitosis. In the G2 phase, the DNA replicated is checked by enzymes and repaired if needed. Now moving on to mitosis. In mitosis, the first phase is prophase. When prophase begins, the nuclear membrane dissolves and the genetic material condenses. The duplicated chromosomes become visible. The spindle fibers originate from the centrioles and extend towards the centromeres of the chromosomes. At the end of the phase, the centromeres all tighten and the mitotic spindle appears at the nuclear and the nuclear envelope disappears. After prophase comes metaphase. Metaphase is marked by the alignment of chromosomes along the metaphase plate and the attachment of the spindle fibers to the centromere of the chromosomes. The third phase of mitosis is anaphase. Anaphase is initiated by the spindle fibers pulling apart the sister chromatids to opposite sides of the cell. The final stage of mitosis is telophase. During this phase, the chromosomes cluster opposite spindle poles, and the chromosomes can no longer be seen and chromatin materials tend to collect. After the nuclear envelope assembles around the chromosome cluster, the cell organelles start to appear. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, but the cell itself is divided into two daughter cells by a separated process called cytokinesis. In the animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cytoplasm into two. At the end of this division, organelles get distributed between the two daughter cells. And now, cancer. The process of replicating DNA and dividing a cell can be described as a series of coordinated events that compose a cell division cycle. At least two types of cell cycle control mechanisms are recognized: a cascade of protein phosphorylations that relay a cell from one stage to the next, and a set of checkpoints that monitor the completion of critical events and delay progression to the next stage if necessary. The first type of control involves a highly regulated kinase family. A second type of cell cycle regulation checkpoint control is more supervisory. It is not an essential part of the cycle progression machinery. Cell cycle checkpoints sense flaws in critical events such as DNA replication and chromosome segregation. When checkpoints are activated, for example, by under-replicated or damaged DNA, signals are relayed to the cell cycle progression machinery. These signals cause a delay in the cycle progression until the damage of the mutation has been averted. The connection between the cell cycle and the cancer is obvious. Cell cycle machinery controls cell proliferation, and cancer is a disease of inappropriate cell proliferation. Fundamentally, all cancers permit the existence of too many cells. However, this cell number excess is linked in a vicious cycle with a reduction in sensitivity to signals that normally tell a cell to adhere, differentiate, or die. This combination of altered properties increases the difficulty of deciphering which changes are primarily responsible for causing cancer. The first genetic alterations shown to contribute to cancer development were gain-of-function mutations. These mutations define a set of oncogenes that are mutant versions of normal cellular proto-oncogenes. The products of proto-oncogenes function in signal transduction pathways that promote cell proliferation. However, transformation by individual oncogenes can be reductant. Mutation of one of several genes will lead to transformation, or can be cell type specific. Mutations will transform some cells but have no effect on others. This suggests that multiple distinct pathways of genetic alteration lead to cancer, but that not all pathways have the same role in each cell type. It now appears that a tumor suppressor gene mutations are highly likely to promote and may even be required for a large number of spontaneous as well as hereditary forms of cancer. Loss of the tumor suppressor gene, product PRB, for example, would be predicted to liberate E2F transcriptional activators without requiring phosphorylation and thus bypass a normal negative regulation controlling entry into the cycle. Loss of tumor suppressor gene product P16 would have a similar consequence by liberating E2Fs by increasing PRB phosphorylation. In addition, cell cycle progression can be halted at several points by the tumor suppressor gene product P53. Activated in response to checkpoints sensing DNA and possibly also chromosome damage, loss of P53 would remove this brake to cycling.